During your course, you had to isolate DNA from plant tissue, and this was done as part of the genetics section of your course. So this video is to help you understand the key stages in this practical, and you might have used onion or kiwi fruits are commonly used in some schools too. So in this video, we're going to discuss using onion. So the first thing we did was we chopped our onion into lots of small pieces. The next step is into a beaker you place some distilled water, some salt and some washing up liquid. So after this you then add your chopped onion to this solution of washing up liquid and salt, and you have to know why you added the salt and why you added the washing up liquid. What exactly are they doing? The addition of washing up liquid is important because the washing up liquid interferes or helps break down those cell membranes and the nuclear membranes, and this releases the DNA from the cell. The addition of salt is important because the salt causes the DNA to clump together, and the reason why you chopped the onion in the first place was to disrupt or break down those cell walls. The beaker was then transferred to a hot water bath, and this was set at 60 degrees Celsius. The reason for this hot temperature was to ensure that those enzymes that would break down DNA are denatured. So after 15 minutes, which was timed precisely because if you leave it longer than 15 minutes the DNA gets broken down, the beaker was removed and placed into an ice bath. So the reason for the ice is to prevent the breakdown of DNA because ice will slow down any chemical reactions that are ongoing, and so this helps to preserve or to prevent the breakdown of the DNA. So this cooling went on for 5 minutes and then the beakers were removed. The contents were then transferred to a blender and blitzed for 3 seconds, so 1, 2, 3. And the reason why you didn't want to blend it too much was you would destroy the DNA. You're blending to further break down those cell walls and cell membranes. So after blending was filtration, and the reason why we needed to filter was to remove the large cellular debris, so the large pieces, and what passed through in the filtrate, the liquid, was the DNA and proteins. So we took some of the filtrate and transferred it into a test tube, and to this we added some protease, and our protease we used was fresh pineapple juice, and the reason why we added protease was to break down those proteins associated with DNA. The final step is add 10 ml of ice cold ethanol to your test tube, and it's usually slid down the side of the test tube using a pipette. The ethanol has to be ice cold because the DNA is insoluble in ice cold ethanol but soluble in ethanol that's at room temperature, and what you see is the DNA coming out of solution sort of floating on the top of the onion filtrate, and you can see that it looks like this slimy white stringy stuff. So that is how you isolated DNA from plant tissue. In our case it was onions. So make sure you know each of the steps but be able to give an account of why you're doing each particular thing. So always remember that doing past examination papers are the key to doing well, and in this video I use some icons from the Down project as credited here. I'm a pro member but I still wish to give credit to the artists.